Hi, I'm Sandra Porter. I'm going to talk today about how we can find protein sequences from new variants of SARS-CoV-2 and how we can compare those protein sequences for the SARS-CoV-2 spike protein to sequences that are found in molecular structure models of the spike protein, and how we can predict whether or not a particular antibody might still be able to bind to a new variant. Before we begin, we're going to take a quick look at one of the antibodies and a portion of the SARS-CoV-2 spike protein. So this is the receptor binding domain here in brown, and bound to it is an antibody that was one of the first commercial antibodies called bamlanivimab. This antibody this is part of the antibody, the fragment that binds to antigen. The pinkish part is the heavy chain, the blue part is the light chain. And if I click the analysis tab here in ICN3D, I'm using ICN3D to look at this, by the way, and I select sequences and annotations, I can also see that same information. So I see here that the pink part is the heavy chain of the Fab, the fragment with antigen binding capabilities, and the blue chain is the light chain. And again, the brown chain is the spike protein. All right, so what we're going to do now is we're going to go to a website that has data analysis and visualization capabilities. This is created by researchers from all over the world who've been studying all kinds of viruses over many years and gathering the sequences and comparing the sequences. What we're going to look at, so we're going to look at SARS-CoV-2, the virus that causes COVID-19, and we are going to look at the latest global analysis that has open data. And the reason we're picking that one is because that's going to allow us to get the protein sequences. So we're going to select that. And what we'll see from next strain is we're going to see a phylogeny that shows us the evolution from 2020 here all the way up through now of the SARS-CoV-2 virus. And you can see these branches show you which sequences are most closely related to each other. And you can see here we have a couple different Omicron branches. We have, in the purple, we have Delta, we've got Gamma, Alpha, Beta. And as I mouse over these, move my cursor over, they expand, so they get larger as I move my mouse. Okay, so I can see a lot of different sequences here. This is actually showing me almost 3,000 genomes, so 2,873 different genomes that were sampled between December 2019 and now. I can see where they've come from in different parts of the world. And different parts of the world are collecting and sequencing genomes. Some are sequencing them more than others, and so that is reflected in the data. And I can scroll down and I can see how diverse different parts of the sequences are. That is, some regions of the virus acquire more mutations than others, and that is reflected in the data here. So here is the spike protein that has that S, and you can see this is a part that has a lot more mutations than other parts of the virus. The reason for that is that the spike protein is the protein that allows the virus to infect cells, as it binds to a receptor on the outside, found on the outside of many different cell types. It's also the protein that is the target of our immune system. And so mutations in this protein can help the virus escape our antibodies. As I scroll down, I can see the different frequencies. These are a little more recent. I can see right now we have a lot more of clade C of Omicron, and we have less of this other clade, 21L, and we do not seem to have much of K or Delta or the other Delta variant anymore. I can also look at these in detail. If I were to click any one of these points, I can see which codon is affected. And if I'll just click this for a moment. It's a little bit tricky, but I clicked that, it expanded the graph, and you can see in red where I've clicked it. And you can see now that at that codon right now, we have mostly it has a D. Okay, D stands for aspartic acid. So right now, 100% of the circulating virus has that particular amino acid at codon 142. Okay. Now, and we can see how that it's changed. When I clicked that, it changed the information in the graphs, and we can see here a lot of this is from clad, this clad here. So most things have aspartic acid at 142. All right, I'm going to go back to where we began. Show all the clads. And I think it's kind of nice, actually, when you look at these, to kind of filter out, and we're just going to look at the emerging lineages. So some of the things that are new. So I'm clicking emerging lineages. And you can see here we've got BA1.1, BA2.3, BA2.12.1, and BA4. And I can see the colors here in the key correspond to the different points in the phylogenetic tree. And if I want to investigate one of these, I can click one of these. I'll just click this one. And I can see this particular one is clade BA.4. This one was found by somebody in, it was from a human in North Carolina. You may be aware that there are also SARS CoV-2 viruses that have been infecting white-tailed deer and minks. So there are other hosts other than Homo sapiens we might be interested in, but this is human. It's found in South Carolina. Looks like it was about a week ago, and this is sequenced by LabCorp. I can also see some information. I can see different nucleotide changes in this sample. I can see some of the different changes in the proteins. And I can actually scroll down and see some of the changes in the spike protein too. So this is one way that I can look at information about this sequence. The other thing I can do is I can click the link to the GenBank accession number, which I'm going to do now. And that is going to take me to the NCBI, which is part of the National Library of Medicine, part of the National Institutes of Health in Bethesda, Maryland. And I can see a genome sequence for that virus. You can see it's about 30,000 bases long, RNA, and this was deposited May 12th, so pretty recently. And now I can scroll down and I can see the protein sequence. What can be challenging with analyzing these is that the protein, SARS translates its protein as a polyprotein. So it translates a whole bunch of proteins together in one sequence. And as a person researching this, that can be a little bit tricky because you would have to find the right part to study the part we want to look at, like the S protein. So there's an easier way to do that. We're going to go to the graphical map. We're going to click graphics. And I've looked at this a lot, but from looking at this, I have learned that this part right here is the part that relates to the spike protein. Okay, so what we're going to do is we want to get the accession number for our spike protein. And here you see it says surface glycoprotein. That's the spike protein. So I'm just going to select that with my cursor, and I'm going to copy it. Okay. And that's the protein sequence I'm going to use to figure out if there are changes in the portion of the spike protein where antibodies bind. Okay, so now what I'm going to do, I've got that sequence. I'm going to go back to ICN3D. And ICN3D now has the BLAST algorithm from NCBI. I'm going to use BLAST to compare the sequence from that variant to the protein sequence here in my structure. Okay, so I'm going to open up the File menu. I'm going to select Align, because that's what BLAST does. It's basic local alignment search tool. And I have a sequence, so I'm going to align the sequence that I got from NCBI to the sequence that is in this particular structure. So sequence to structure, and I paste my sequence here, the accession number for my sequence. What will happen is that ICN3D will use that accession number to get the sequence from the NCBI when it does the comparison. Now, the other thing I need to do here that's really critical is I need to put in the ID for my structure. So you see here it says 1 TSRA, but that's not the structure I want to compare anything to. Okay, the structure I want to compare the spike protein to is the spike protein in my structure, which is 7kmg_C. C is the name of the chain name. So now I have 7kmg_C, and then this is the protein accession number for the spike protein that I got from NCBI. Okay, so now I'm going to say align with BLAST. And you can see here, this is the structure showing where the changes are in the receptor binding domain of the SARS CoV-2 spike protein. The blue spots are places where the amino acids are different. The pink spots or lighter blue spots, they're different, but maybe not quite so much. Pink spots, they might be a little bit different. And we can see here over on the right-hand side is the alignment. So the structure, the sequence from the structure is at the top, 7kmg_C. The sequence that I got from the NCBI is here, UK 32191.1. And the alignment is shown over here with the sequence. And wherever there's a change, you can see there's a gap here, and you can see where the change is in the alignment. All right. Well, now what I'd like to know about is I'd like to know, okay, there's some regions that changed, but are those regions that have any kind of relationship to where the antibodies were binding? Are there changes in the antibody binding site? So now what I'm going to do is I'm going to get my antibody sequences back. I'm going to open up sequences and annotations. Actually, I'm going to go back to here and say show all chains. There we go. Now I can see what the antibody sequences were again. And what I'm going to do now is I'm going to see where's my antibody binding to the spike protein. And I'm going to add an annotation so I can find that site more easily. So what I'm going to do, I'm going to open up the select menu, and I'm going to select by distance. What I can do here is I can find all the amino acids that are within a certain distance of my antibodies, which means they're close, right? It could be part of the binding site. So I pick by distance. I'm going to stretch this out a little bit more. I just drag the corner to do that. And I can stretch these out a little more by dragging the corner also. Okay, so what I want to do is select my antibody. So that's going to be chain A, which is the heavy chain. And now I'm going to have to hold my shift key down, and I'm going to select chain B, which is the light chain. So now I've selected the antibody, and I want to see the region of the spike protein that is close to the antibody. The antibody, that spike protein, that was 7kmg_C. So I'll click that. And now I'm going to click display. And what has happened now is it's highlighted the antibody binding site in the structure and in the sequence as well. Now what can be bad sometimes is that if I'm not careful, I can click something by mistake and those highlights go away, and then I have to do this all again. So what I'm going to do now is I'm going to save my selection. I can just get rid of that. I'm going to say select, and I'm going to say save selection, and I'm going to call it antibody underscore binding site. There we go. So now I have the antibody binding site annotated, and which means that I can find it again really easily just by selecting it in my list of structures. Okay, so now I have the antibody binding site highlighted. The question, I might go back to my other question, is what changes took place in the antibody binding site? I'm going to stretch this out so I have a bigger view of my sequence. And let's take a look. We can actually, I'll just kind of scroll through here. So remember, I've got the protein sequence up on top, and I can scroll through this. All right. So the yellow the highlighted amino acids, those are part of my antibody binding site. There are some changes that are close to those amino acids. Those might be important, but for now, let's just look at what's in the antibody binding site itself. And as I scroll along, I can see that there's a change here where there is an E at position 484. So that is a glutamic acid at 484, and it gets replaced by an alanine. All right, that could be important. I see there's also a change here, and looks like that's the only other change that I'm seeing directly in the antibody binding site. Okay, so I'm going to look at this glutamic acid at 484. I mean, that could be important, right? So here's how I'm going to do that. What I can do now is I can go to analysis, and I can model the effects of that mutation and see how that mutation may change the interactions between the two proteins, the different proteins. So I pick mutation, and I'm going to describe my mutation here. It was at 484. It's in chain 7kmg underscore C, 7kmg underscore C. And it was a, so I've got the protein PDB ID, but 7kmg. I've got the chain ID here, which is underscore C. Now the residue number was 484. All right, this is a key I'm using here to show me what to put in here. And the mutation is an alanine. So I'm putting in an alanine. All right, there we go. 7kmg, the chain name, the position number, and the change. And now I'm going to ask to look at interactions. Okay. So I selected interactions, and it shows me a prediction of, one, the interactions that normally take place, and two, the interactions, how the interactions would change, or if there'd be any new interactions that would take place if I've with my mutation. Okay, so I can use the alternate key, or I can just click this button to see how this varies in the three-dimensional structure, because right now they're superimposed on each other. So I can click that, or I can use the A key on my keyboard and see how it changes. All right, So I'm going to turn this a little bit so you can see this a little better. So here is the glutamic acid. The glutamic acid, remember, is part of the spike protein. So here's where it's connected to the backbone, and here sees the amino acid sticking out. And glutamic acid has a negative charge, and it's got a carboxyl group at the end here. So these are oxygens can have a negative charge. All right. So normally, if I look at this, you can see that it's forming a lot of interactions with other amino acids, both on the blue light chain and on the pink heavy chain. And I have a key here to tell me what those interactions are. So the green represents hydrogen bonds. So there's a hydrogen bond here and here and here. Okay. The cyan color, which is kind of a turquoise, represents a salt bridge. So between a positively charged amino acid over here or here and our carboxyl group on the glutamic acid. And then we've got gray, those are contacts, different kinds of contacts or hydrophobic interactions. There's no halogen bonds here or other kinds of things. Okay, so there are a lot of interactions that you can see normally between the glutamic acid and the heavy chain and the light chain. And these are all detailed over here in this diagram. So here's the glutamic acid. Here is an arginine at position 50 in the antibody, the heavy chain antibody. Here's other interactions in the heavy chain. And here's an arginine in the light chain of the antibody. And then this amino acid also interacts with amino acids within the spike protein itself. Okay, so now you can see it has a lot of interactions. If I click the A key and alternate between the structure models, here's our prediction for what happens when that glutamic acid gets replaced by an alanine. So now a lot of those interactions have disappeared. There's still, it looks like, a possible hydrogen bond here, maybe some other interactions, but alanine's a lot shorter. There's a lot fewer interactions, as you can see. We alternate. As I can also scroll down, and IC in 3D will show me the differences. It'll show me the things that are common and stay the same between the two structures. And it shows me what happens with the mutation, what things are different. So with our original structure, we see that we had salt bridges and some other contacts that are different. With our new structure, we've lost all of those. We don't have any really new interactions. So now, not only do we know that we had that amino acid change, right? Here, back to my amino acid binding site. Not only do we know we have this change here at 484 that happens right within the amino acid, right within the antibody binding site, we know that that change is probably an important change because it's disrupting a lot of really important strong interactions. And that's all.